morning, everybody. Welcome to the first IIDR combined rounds for 2024-25. I'm Laurie Burrows. I'm the Associate Director of the IIDR, and I'm the moderator for these conversations. So what we're going to do is we're going to have two presentations back to back, and then we're going to answer questions at the end. So if you have questions through the session, just type them in the Q&A box, and then I'll moderate them at the end. If we're getting short on time, then I'll ask the speakers to just write their answer back to you so that you don't get left hanging without an answer. So today, I'm very happy to introduce our two speakers, Andrew McArthur and Rubayet Hasan. So Andrew is the David Braley Chair in Computational Biology at McMaster University and a professor in the Department of Biochemistry and Biomedical Sciences. He's a member of the IIDR, and his research team focuses on building tools, databases, and algorithms for the genomic surveillance of infectious pathogens. One of these databases is the widely used Comprehensive Antibiotic Resistance Database, better known as CARD, and its clever subcards like Card Shark, which I love. Today, Andrew will be speaking to us about genomic surveillance from a data science and design perspective. Our second speaker is Rubayet Hasan. He is a clinical microbiologist and an assistant professor in the Department of Pathology and Molecular Medicine here at McMaster. His current research focuses on the application of novel technologies, including next-gen sequencing, to the diagnosis and surveillance of infectious diseases. And today he'll be speaking to us about the practical challenges for genomic technologies in a clinical setting. So thank you both for speaking to us today, and I will hand it over to Andrew.
I'm going to, honestly, I'm going to try and set Rubayet for your talk. One of the cool things, I've been at Mac 10 years, coming here with this institute is the opportunity for someone like me who's a data scientist person and a genomicist to really work towards the clinical point of the spear. It's not something I really planned on. But the opportunities to collaborate have been fantastic. And so it's good we're giving this talk together because we're just starting to collaborate. So I'm going to talk a little bit about molecular epidemiology and some methods and things that worry us. So there's three perspectives, right? You can, starting at the right, you can work at the national or regional, international level. So this is really the surveillance, and this is the meat and potatoes of what my lab does. On the far left, you have the idea of the patients and doing diagnostic on individual patients. And then in between is questions around the local population, which is really around typing and outbreaks to know what's going on in your community. So, on the national side, we run this CARD database. It enables public health agencies to figure out what resistance genes and mutations are in their bacterial pathogens. And so we collect that data, and you can look at trends, internationally. On the patient side, DNA sequencing is starting to make inroads. There's a practical, it's not that affordable yet. So patient diagnostics are primarily assays. They may be based on genomic data like PCR, but a PCR reaction or a lateral flow like a COVID test can get you a ton of information. So sequencing has its place when it can add value beyond existing diagnostics or even just help design new diagnostics. But where sequencing at the sort of the clinical level is really starting to make inroads is around typing in outbreak. So I'm just going to highlight one of our studies from COVID pandemic. So this is a Hamilton hospital that had an outbreak, which is just a defined increase in numbers on 5 wards in the hospital. It included both patients and staff. And what we did is take the swabs and sequence those genomes of those. So what you don't know when your numbers go up, if this is just bad luck, that these are all independent cases coming in from the community on the ward, or whether you actually have transmission going on in the ward. And your infection control response has to be very different from that. So by sequencing, the first thing on the top left, we noticed that there was more than one SARS lineage in here. So even if there was transmission going on the ward, there were independent events going on. Not everybody was involved in a possible true outbreak scenario to it. So that suggested that this was probably complicated. On the right, though, the one type, the B1564.1, was the more common. So a lot of them had this exact type. And typing only tells you that. It doesn't really tell you if the transmission, but the advantage of sequencing whole genome is you get down to the individual nucleotides. So you get this figure on the right, where green is all the community and other sequences from around the globe of this particular type, but blue is our outbreak. The size of the circle is the number of patients with the subtype. So what you saw is a large number of patients and staff had the identical type down to the nucleotide, not a single nucleotide difference. And then there were 1 or 2 mutation differences. So that is strongly suggested that it is transmission in ward. Mutations occur fairly quickly in SARS in these settings. So this is your first hint. There wasn't enough clinical metadata to really say, who was transmission and what are the patterns, but this was strongly indicative of it. The trouble with this was still retrospective. Our sequencing team was not up to a 48-hour turnaround on this type of thing. By the end of the pandemic, we could probably do that. We could turn around and actually help in real time. So I am coming from a design and data science perspective. So there's really 3 revolutions. I used to talk about 2, but really now there's 3. The first is the internet. So I picked this screenshot because I got my PhD in '96. So when I started my PhD, GenBank arrived as a CD-ROM to your lab. And then by the end of my PhD, there was a website you could use to look at nucleotides. So that revolution of that, and technology is just constantly improving, and it gives us advantages. The other thing that's occurred is the change in DNA sequencing. So this is an S35 acrylamide gel. Again, that's how I sequenced back in those days. Now we're highly robotic. And then the big revolution is the pandemic really brought sequencing. It's the first time we basically watched a virus evolve in real time. And my lab used to think we were 5 years ahead of sequencing really having a strong place at the local hospital. And now we're 5 years behind because every local hospital has a degree of sequencing capacity because of the pandemic. So if you talk about the internet, that's one of my favorite websites. This is all the undersea cables that transmit data for the internet. So that tells you how a connected world to it. So this allows us to share data. So you may be working on a single patient in Hamilton, but allows you to put in context the databases, all the related strains out there, helps you figure out travel history, figures out that has unique resistance. And there's policy aspects. So at this stage, it's almost impossible to publish a paper with sequence data without submitting your data to a database. So this is microbial genomics and access microbiology. They say, we will not accept your paper if your data is not in a database. So this is good. That means data is almost forced to be shared. The trouble from a design perspective is the amount of data. So again, you might be working with a single patient, but the reference you want to compare of it can be massive. So this is a chart in the increase of finished genomes versus whole genome shotgun sequencing in GenBank, one of the biggest databases. And that inflection point just after 2000 is when next generation sequencing really took off. We see a skyrocket in this type of data. So while we might do small-scale sequencing on an outbreak or a single patient, the reference data is almost to the point of getting unworkable. And the reason for that is the other revolution is that sequencing is a very competitive market. There are many technologies, and the short thing is it's getting faster, smaller, cheaper, and higher throughput all the time. So that means what you see sequencers go from large research institutions to now having a sequencer down at the local hospital. And that hospital at reasonable price can generate a very large amount of data. And this is just accelerating and get worse and worse from a data science perspective. Huge benefits for wanting to learn things and to make good clinical or research decisions, real problems when it comes to data science. And again, the pandemic is what really threw this to the new level. So you had GridION nanopore technologies, or Illumina were the dominant sequencing platforms. Here at McMaster, we used Illumina. But if you looked at Ontario Institute for Cancer Research, they used the nanopore GridION platform. But what happened is we basically sequenced variants in real time. In Hamilton area, with the provincial reporting that we were part of, our turnaround time was 7 days from swab to submission of the data. And this is how you got your weekly report of what variants were in the community. So with that background of these 3 things going on, from a data science perspective, we're going to go back to computer science. So Moore's law is something we depend on in comp sci. And this is empirical data that computing capacity of chips doubles about every 2 years. So that means 2 years from now, you can spend about the same amount of money and get twice as much computing power. And we see that industries that are following Moore's law or behind Moore's law are very workable. right? The computing's always getting ahead of them. But this is the Carlson curve. This is the cost of megabase of DNA sequence, which has plummeted since next generation sequencing came along. And so it is well below Moore's law, which means the rate of which we produce sequencing data is outstripping the ability of computing to keep up, which means your overhead for buying chips just to keep up with the data you want to produce is huge. You're going to be constantly spending money on chips. I couldn't find newer data, but this is Kreider's law. So the capacity of hard drives, spinning drives, doubles roughly every 13 months. In pre-next gen, we considered hard drives trivial. We didn't spend that much. They were cheap. We never produced that much data, but next gen sequencing went beyond Kreider's law. And so people that use our high performance cluster and in-house science know that we spend $40, $50, $100,000 a year just on hard drives from all the people doing the work. So again, if sequencing gets to your local hospital, how much budget do they have to keep up with their data, particularly if they're probably mandated to keep that data for a large number of years? The last one is Butter's law. This is that the data coming out of fiber doubles every 9 months. And the good news here is that genomics hasn't passed Butter's law. There's Netflix is our big friend here. They keep octable capacity going up. But the trouble is the fiber's in the road. The cables that are in the wall and going to your sequencing core might be older cables and switches. And so while if you can get the data to the street, you're in good place. But often, and we see this in Hamilton facilities, it's getting the data to the street from inside your building is the problem. And again, if it's a massive database on the other side, you need to access, this becomes a real problem. So I'm going to talk about a few usage cases for that. So I'm going to start with wastewater as a proxy for population biology. So this is increasingly popular. You see in COVID, we use wastewater because this is the outfall from a community. So we're looking at community sampling. But federally, we are starting to sequence wastewater to track AMR genes, for example. There's a proposal going on, a major federal effort. The trouble with this is you're doing what's called metagenomic sequencing. You're sequencing all the DNA in the sample. So in this case, I put a cartoon for 10 million sequencing reads, which is no small amount of effort or money. But most of it is from the humans. It's from the bacteria that live in a wastewater processing plant, the natural microbiome, et cetera. And the DNA of interest is a tiny fraction. So your target is a needle in a haystack, and it takes a lot of expensive sequencing to find it. Also with metagenomics, the data is very fragmentary. You're not getting, here's the E. coli genome from individual A at 243 Hunter Street. And because of this, you have a huge storage and computing need to this. And this is a big focus on my lab, is how to get through this, how to reduce the pressure so this is more affordable and less of a burden to do this work because the data is of high value. So I'm just going to compliment PhD student Dirk Hackenberger, who is shared between the Poinar Wright and MacArthur labs. Poor guy, he's got 3 supervisors. But Dirk took an early platform we built, and he built what was called a bait capture platform for metagenomics. So what bait capture is that you make molecular fish hooks to target specific DNA. So at the top left here, we have a sample, and only one out of the 5 molecules we want is our target. You make molecular fish hooks that only bind that green target, and you use magnetic beads to then pull down that target from the molecular fishing hook. And what washes out is all the non-target DNA. So what's left in your sample is highly enriched for the DNA you were interested in. And so bait capture was used. Henrik Poinar here is famous for it, for getting ancient pathogens out of archaeological samples. But what allows you then that your sequencing effort is, instead of being the minority the thing you're after, it's now the majority the thing you're after. So Dirk's idea was that probe design is difficult, and in particular, we wanted to build a probe set for AMR, which changes on a regular frequency. There's always new variants, and as such, there was never an up-to-date probe set, and then it was really expensive to make these probes. So he wrote software to use our CARD database, which regularly updates with every variant and gene. So every time the database updates, the software would design a new bait set. He experimentally validated it, but he became really innovative. He came up with an in-house synthesis protocol that reduced the cost by 2 orders of magnitude. So this made it very effective. So this is some work from wastewater. So he produced 4 different types of the library in his experiment. But if you look at this graph, it's the green line at the very bottom. This is unenriched metagenomic sequencing. So up to 3 million sequencing reads, which again is not a small amount of effort. And without enrichment, because the target is that needle and haystack, they barely detected any AMR genes at all. But the four curves you see is where we use different enrichment platforms and molecular fish hooks, we start to see hundreds of AMR genes in the sample. With those detections, he was seeing in wastewater samples an enrichment from anywhere from about 300 to 600-fold increase in detection levels. What that means is not only do you get high detection, but you can sequence less to get that detection. So it's less money, it's less effort to get more data and more information. So this was really excellent work by Dirk and his collaborative team. But from the bioinformatics side, there's an inherent problem in these data, and it's something unique to bacteria, can be very unique in virus as well, but not something you see in humans. So these are the TM beta-lactamases, these metabolized beta-lactam antibiotics, and there are hundreds of variants of this. And some of them are only 1 or 2 nucleotides different in their genetic code. So they're very similar. So when you get the fragmented sequencing data from Dirk's experiment, you want to figure out, well, which TEM did I see in that wastewater sample? The algorithms that align fragmentary sequence data to reference are from human genomics. And so the most common you'll see used in the literature, even if it's AMR work in wastewater or soil, is something called Bowtie2. And Bowtie2 assumes you do not have an allele network, that you have hundreds of variants of a single gene. And so what Amos in my lab did is he simulated sequencing of 7 AMR genes in a metagenomic sample. And then he compared them using Bowtie2 to the reference database. Instead of getting 7 possible results, he got well over a dozen possible results. And what the algorithm does, because it's never thought about allele networks, is it can figure out that it's dealing with, say, a CTXM, but then to figure out which is specific one, it almost starts to guess. And so you get low coverage of a whole bunch of variants, and it's not making a call on the variant. So this is a bioinformatics challenge. Our colleagues in Denmark came up with what's called the KMA algorithm. It's a version of this that's designed for allelic networks like AMR. And you take the same data set and you find 7 genes. And so you simulated 7, you find 7 in the data. 2 of them are actually the wrong variant of the gene, so there's still a limit to here. So this is an advance that we get much more precise data. And frankly, if you're reading the literature about AMR in soil or water or wastewater where they're using Bowtie2 and metagenomics, you should be very suspicious of those results. Trouble is, this algorithm takes more math to do this. And so there's a computational load to get it done with a large data set. So we've been working on trying to push this space because Dirk made it cheaper to do wastewater. So the nice thing is wastewater is essentially a complex sample, and you're trying to pull useful information on a complex sample. And that's very analogous to a clinical swab. So this is a swab we use with our rectal swab we use in our babies in the NICU. So that swab similarly have a lot of host DNA, lots of natural microbiome, and maybe the target pathogen or gene that you're interested in. So these bait capture approaches can be applied. So this is a paper that's in review. This is the Jarovensky Cancer Ward. Routinely, there are swabs done for VRE and MRSA, so infectious pathogens. So we piggybacked on that to see if there was asymptomatic C. difficile carriage. And there was a quite high carriage. So these are patients that have no symptoms at all, but they are carrying C. difficile. And we cultured them and we sequenced those genomes. And what you could find when you looked at the epi data combined, that you had actual patient-to-patient transmission asymptomatically happening in the cancer ward. Of course, you treat with antibiotics or anti-cancers, you could then go into becoming a symptomatic patient. So this was hugely informative about the background transmission and carriage rates of C. difficile in patients. The trouble is it was a retrospective study. We had to culture from these swabs, which takes effort, and then the extra cloning and sequencing. So again, this is not a real-time tool. This is where Maddie McCarthy in my lab comes. This is a PhD student in my lab. So we've talked about bait capture enriching a complex sample. So she's developing bioinformatics pipelines that will look at every known variant of C. difficile, whether it infects a human, a horse, a dog, you name it. Because you can't just build bait capture for one isolate, because you don't know which isolate is going to come into your community and your patient. So she analyzes tens of thousands of genomes to design a bait set that will be efficient and cost affordable for any isolate infecting a patient. And then her intention then is to do bait capture from swabs, so you bypass culture. She's literally in the stages of doing the validation experiment. So ask us in a couple of weeks and we'll tell you how well it worked. So it's very close. But Maddie has ambitions. This works. There's still a lot of labor. This is an Illumina sequencing platform. There are limitations to that. She wants to bypass this. So she wants to bypass big culture and get rid of those extra steps, those extra reagents and cost. So our target is a nanopore sequencer. So this is their tiny version, a MinION that plugs into your laptop. This is a newer sequencing technology that has a little nanopore that you take the DNA, very little preparation to it, on handling, just essentially DNA extraction. And there's many pores sitting on this sequencer, and one DNA molecule goes through each pore, and it gets a long sequencing read. So this is great technology. The reagent overhead is smaller. The hands involved in prep is less. But it will sequence all the DNA. So again, the data that comes out would be mainly the human in the swab. But there is an algorithm called adaptive sequencing where you can train the pore in the machine to test the DNA as it's being sequenced and say, I'm only after C. diff. And if the sequence I'm looking at is not C. diff, it reverses the polarity and spits the DNA out in real time and grabs another DNA molecule. So it's enriching by algorithm in this case. It's a brilliant little approach. So what Maddie's doing is we routinely use swabs to scan for VRE and MRSA, and she's adding C. difficile. So she's trying to train the nanopore sequencer in real time to only sequence these 3 pathogens from a clinical swab. It will spit out all the DNA from the normal microbiome and from the patient genome. So the idea now, we've bypassed the bait capture. We don't have to synthesize baits and all the extra reagents, but also this is not Illumina. This is a faster sequencer. This is getting close to rapid response if the in silico adaptive sequencing enrichment works. So she's just now setting up experiments to validate this approach. Again, you probably wouldn't use it all the time, but as a surveillance tool, it could be quite useful. The last little bit is scale, to come back to the computer science. So this is Signal. PhD student Jaylee's Nasir on the left led the national consortium to build this. This is a pipeline for high throughput sequencing of COVID variants using Illumina. It was a pandemic software. It's designed for doing tens of thousands of isolates a day. It is the national software for FAC, for the Public Health Agency in Canada. This is the national platform. It's used every week in Hamilton Health Science for our COVID reporting. This thing follows all the standards. It's powerful. It does everything you want for rapidly screening variants, but it's a monster. It takes very serious computing for this to do well. That it's a strain for a local public health agency to maintain this, to keep this. And we get donations from Cisco and Hewlett Packard to do this. So this is the last slide I'm going to talk about. There's an issue of accessibility and equity. Sequencing alone is a technology that everyone can access, but if they can access, they may not be able to access sufficient compute. And as I put out, it's just going to get worse year to year to keep up with the CPUs and the storage, et cetera. So we started collaboration with the Chan Zuckerberg Foundation. They have a metagenomics platform that's free for use for researchers and public health agencies around the world. And they've added all our AMR algorithms to it. So this allows researchers to not have to maintain their own computational cluster. All they can do is focus their effort on building their sequencing platforms in-house, and this takes care of it. I think Rubayet could probably bring up there's issues of confidentiality and data that you have to work on, and not all cases can use this, but equity is a really important thing. So I'll stop there and thank all my crew and collaborators, a lot of collaborators on this call for this, and I'm going to hand it over.
Thank you so much, Andrew. You did the hard part. Let me share my screen first. All right, as I was saying, you did the hard part, and thanks to your work, that actually solves half the problem we generate in the lab. And for the audience, I just want to say that, at the end of my talk, you'd probably see how our conclusions actually converge, and despite the fact that we both are probably working in a completely different setting independently. So my objective today is very briefly review the limitations of the existing diagnostic tests for infectious diseases. And I guess Andrew has covered most of the sequencing technology part, so I'd probably skip. And then I would discuss what is the appropriate context when DNA sequencing should be applied, and discuss some challenges and potential solutions based on our experiences. So first of all, you know that our existing standard diagnostic test for infectious disease actually works under 5 main principles, among which microscopy and culture, which is like known for more than 100 years, and but is still probably playing the most important role in infectious disease diagnostics. The antibody detection mostly makes retrospective diagnosis, and antigen detection has a very limited availability. On the other hand, the detection of nucleic acids or NATs are actually an incredible addition to our existing diagnostics, offering high sensitivity, specificity, rapid turnaround time. And this is all like thanks to the 50 years of advancement in molecular detection techniques in actually all fronts, like, starting from DNA hybridization to amplification to sequencing. We have witnessed a massive transition over the last few decades. And I think you probably have seen a very similar picture Andrew has presented. And I just wanted to, say you that, I've been using this diagram in my presentations in the last 10 years. I have changed this figure like about 3 to 4 times, and every time I change, there's like one more billion or few more billion whole genome sequences added to the database. So this is important for us in microbiology because our knowledge about the genomes of pathogens is very important in terms of how we utilize DNA sequencing for infectious disease diagnostics. And because of these massive advancements, I think molecular testing is now considered to be one of the fastest growing field in microbiology. And in terms of prediction, especially after COVID and the massive use of molecular testing, I think it is predicted to reach up to 50 billion market by the year 2032. And however, I think in terms of molecular testing, as you know, most of the infectious disease tests are actually qPCR based, probe-based qPCR based. And here I put some, existing commercial platforms. I mean, they're available in various formats as syndromic panels in a high throughput setting or a point of care setting. In addition, even the laboratory developed tests used for infectious disease diagnostics are actually mostly qPCR based. Although, the history of DNA sequencing is actually much older than PCR. If you look at the years, these two gentlemen got their Nobel Prize, like Frederick Sanger, who actually got double Nobel Prize in 1980 for DNA sequencing, and then Kerry Mullis in 1993 for in chemistry. Despite that, the role of DNA sequencing in infectious disease diagnosis actually still remains very limited compared to, if you think about other areas of laboratory medicine in cancer diagnostics or genetic defects. So, and in the last 2 decades, we also saw a revolution in evolution of sequencing technology. I think Andrew has already talked about it. I'm not going through much details, but you can see the difference in the capacity, starting from like somewhere when per gel or per machine could generate 10,000 bases of DNA. Now we're talking about billions to trillions of DNA bases, per machine or per slide. And as an example, if you think about the first human genome sequencing, it took actually 13 years to complete at a cost of 2.7 billion, and with involving participation of 20 laboratories across the world. And compared to that today, a human genome can actually be sequenced just in a matter of days with a cost of $1,000. So Now, why do we need DNA sequencing for infectious disease diagnostics? To answer this question, I think we need to look at the logic how diagnostic microbiology works. So an infected human body actually generates two main types of specimens for us. One is the non-sterile sites, such as upper respiratory tract or gastrointestinal tract, which is loaded with normal flora. So for this type of a specimen, what we want to do is we want to test with a hypothesis, or we want to target specific pathogens. That's why we design or run a respiratory PCR panel or GI panel or CT-NG PCR for genital specimens. But even if we use culture, we don't look for every organism there. We only look for specific pathogen. So these are targeted tests tested with a hypothesis. On the other hand, we also have sterile site specimens like blood, CSF, and tissues, which are supposed to be germ-free. So that's why, even in this type of specimen, even the presence of apparently harmless bacteria could have a serious clinical significance. So that's why we want to be unbiased for this type of a specimen. So that's why we try to use non-targeted or partially targeted tests, and microscopy and cultures are kind of examples, and sometimes we even enrich samples so that we allow any organism to grow in this kind of scenarios. Now, the problem with microscopy and culture is that there are some limitations. So the limitations is that, first of all, microscopy has poor sensitivity and would not be able to see everything under a microscope. Culture, the problem is, some organism may grow extremely slowly, or some organism may not even grow in the artificial culture conditions that we use in the laboratory. So, and then there are sometimes the bugs are already dead because of prior antibiotic treatment. And PCR is great in terms of sensitivity and specificity, but it is limited to only common and target pathogens only. Then there are emerging, re-emerging, and travel-associated pathogens for which our labs are not always prepared for. And although for antimicrobial resistance, we do have standard phenotypic susceptibility test, there are certain type of antibiotic resistance in certain type of organisms that we cannot test phenotypically at this moment. So as a result, actually is a significant proportion of sterile site infections, such as meningitis, encephalitis, or osteomyelitis, or infective endocarditis remains unrecognized. Now, what does DNA sequencing offer then? So we hope that at least theoretically DNA sequencing can overcome some of the limitations of culture methods. Say, for example, the DNA sequencing should not discriminate between live and dead bugs. As long as there is some DNA from the genome, we should be able to detect them. And then DNA sequencing is not also limited to the growth of organisms that are difficult to grow. So we should be able to detect them by DNA sequencing. In addition to pathogen detection, the DNA sequencing should also give us additional information, such as antibiotic resistance or viral drug resistance. Sometimes we may need to know the virulence determinants in the organisms, pathogen typing for infection control and contact tracing, as well as DNA sequencing may generate valuable epidemiological data that are important in infectious disease management. So in terms of the application of DNA sequencing in clinical microbiology, as I mentioned, it is quite limited. I think the most important one is the 16S rRNA gene or fungal 18S or internal transcript sequence sequencing, which is known for actually almost for 4 to 5 decades. And this sequencing you can use both for sequencing bacterial or fungal isolates that we grow in the laboratory, but we are not able to identify. But they can also be used on the direct specimens. But these are not currently done routinely by all laboratories, mostly offered by the reference laboratories. And also there's no commercial IVDs for these. I mean, most of this work is done in the laboratory test developed test format. So sequencing-based commercial IVDs are actually extremely rare, probably other than a couple of rare examples of HIV drug resistance assays. And commercial metagenomic service providers are also very limited. To my knowledge right now, like UCSF offers a metagenomic service for CSF and plasma, and the cell-free DNA sequencing by a company called Carriers. And Arup Lab also offers a test called CMV drug resistance. So the reason we are interested about 16S or 18S for bacterial and fungal detection is that it lies within actually the primary structure of their ribosomal RNA genes. So this gave us an unique opportunity because they're, they have this alternating highly conserved areas and hypervariable regions. And it enabled us, to design primers from the conserved regions so that we can amplify and sequence by Sanger sequencing. So similarly, eukaryotic ribosomal RNA genes, mainly 18S ribosomal RNA genes, also has an internal transcribed region which has been used for Sanger sequencing. There are also other targets, but these two are the mostly used ones. So in 16S gene sequencing, what we do is either we get an isolate or we extract nucleic acid from the crude specimen. We then amplify, with the 16S primers to generate, say, approximately somewhere from 500, about 500 base pair of sequence. And then we run a gel if the product looks clean, and if there is a product, we then subject them to Sanger sequencing. Nowadays, they are kind of automated, capillary gel-based sequencing. And then they generate, if successful, they'll generate this kind of sequence. So, and the bioinformatics for that is quite simple as well. You just take that stretch of DNA and blast it go to NCBI, blast it against the database, and look for homology with a known genome sequence there. And CLSI actually has a criteria, how do you interpret these results, and when you can actually call a result species specific or genus specific results. So just in terms of clinical impact of running 16S sequencing, I just want to give an example. Most of the time 16S is actually successful in situations with a high pretest probability. Say sometimes you see things by gram staining, but is unable to grow. It is more likely that the 16S would be successful in those scenarios. Or if the patient has high risk with annual contact or travel associated infection, but which our existing diagnostics is unable to identify, in those scenarios, sometimes 16S is useful. I just give you an example that we are experienced with. We had a heart valve tissue from a patient with a significant animal contact, and then the patient had culture negative endocarditis. So we ran 16S and we identified Bartonella quintana in that specimen. And this had a direct impact on patient management and a change in the antimicrobial management. However, there are a lot of limitations of Sanger sequencing. So first of all, Sanger would not work if your sample is contaminated or mixed. I just want to give you an example. In our lab here at HRLP Virology Lab, we have run 16S sequencing on 64 pleural fluid, synovial fluid, and tissue specimen, of which 21 was positive. And 9 out of 29 generated mixed bases. Although in the gel, you could see like a clean band like this, but when you sequence them, you see there are like a high proportion of mixed bases, which as a result, the identification fails in those scenarios. 16S also has a kind of poor sensitivity. When you know a target in general, you would not want to sequence anything which has a PCSCT over 30. Sometimes there are risk of misidentification because of poor sequence quality. And because we rely on a very small stretch of DNA, they may have a poor resolution as well. In many cases, you may not be able to call the results at the species level. And 16S doesn't universally work well for all bacterial species. For some bacteria, other amplification may be suitable. And obviously the 16S is quite lengthy and cumbersome as well in terms of workflow. And furthermore, an important component of part of our infectious disease diagnostics, the viruses and parasites cannot be identified with 16S. So about a decade ago, we, along with a few other laboratories in the world, actually, we hypothesized that, when, the whole world was suffering from genomic fever with the advancement of next generation sequencing, we were thinking, how can we apply NGS, for infectious disease diagnostics, and whether, it is useful to apply this technology, especially in cases of sterile site infections, and that may have been previously unrecognized by the existing test. So in that way, that is now termed as clinical metagenomics, which means basically do the comprehensive analysis of microbial and host genetic material in samples and try to identify the patients bioinformatically. So a few years ago, we have completed this study. It was a validation of metagenomic based study to run on CSF in hospital acute care setting. So first we established a Illumina MySeq based protocol to run one sample per MySeq flow cell. And we established the laboratory part, as well as we validated a bioinformatic pipeline. I think the Chan Zuckerberg, the cloud platform, I think Andrew already mentioned about it. We were actually one of their very early collaborators while trying to do this project. So we also tried several other bioinformatic platforms. So we had either issues with sensitivity or specificity. But with this platform, by applying, after the analysis, by applying a specific filter set, we were able to optimize the specificity of the assay by using a training set of spiked specimens. And with the same simulated specimens, we estimated a limit of detection about less than 1 CFU per ml, and the correlation with PCR-CT were very good. The reproducibility by pathogen type based on NGS reads ranged from somewhere from 2 to 11%. Now, about the validation study, it was a retrospective validation study. The population was were the children in British Columbia who were suspected to have CNS infection. Their age ranged from 0 to 18, with about 68% males. The patient specimens came from various locations, from ED, ICU, and surgical units. And so we tested a total of 74 CSF samples, of which culture and or PCR positive were 28, 37.8%. But, a lot of them were enterovirus positive because, enterovirus meningitis is one of the most common viral infection in children. But we don't expect to detect enterovirus because it is RNA virus. So if we exclude that, our positive percent was about 25.7%. We actually got a really good sensitivity and specificity in that study with 100% and 95% and overall 96% accuracy. So there are 3 pathogens that were additionally detected by metagenomic NGS. But 2 of them were group B strep, which was seen in gram stain but was negative by culture. So but in that way, the group B strep, unfortunately, we did not have a full meningitis PCR panel at that time. Otherwise, I would assume, group B strep would have been picked by the PCR. We didn't really need to do metagenomic NGS. So I think in terms of, although the performance looked good, in terms of diagnostic yield, I mean, it still didn't seem worth to do this, test, because we did not, detect a significant number of additional positives with potential clinical impact. So at this stage, we changed our hypothesis a little bit here. So we wanted to, first we wanted to expand the study, not only to CSF, but also to other sterile site specimens, such as, joint fluid, tissues, or pleural fluid. And then instead of testing all samples, we wanted to test with a strict referral criteria, including that the specimen need to be negative by all conventional tests, And then, we actually listed out a number of criteria so that the pre-test probability of infectious origin, could be, of infection could be increased. And then we also wanted to assess the clinical impact of the test. So this was the study was done in Qatar, in a hospital, Sidra Medicine, which was funded by Qatar National Research Fund. First, we needed to optimize our assay for other specimen types. So we again generated a bunch of simulated specimens like whole blood, tissue, pleural fluid, pericardial fluid, and synovial fluid. And we spiked a representative bacterial viral species into each specimen. And in terms of agreement with PCR, I would say they were pretty good. And in cases when it is not very good, like pericardial fluid, it is because additional pathogens were detected by metagenomic NGS. So in terms of correlation with PCR CT values, majority of them were significantly correlated. The two samples, tissue and pericardial fluid, tissue and synovial fluid, they are not significantly different. And these are also two actually problematic specimens for metagenomic sequencing. I'll discuss a little more later. So all of this sequencing were actually we did on the Illumina MiSeq using the NextEra XT library kit. So we took about 17 retrospective specimens that were culture positive or negative. And after metagenomic NGS, the world agreement was about 82%. Metagenomic NGS generated a couple of interesting results compared to the culture. Like in one sample, we were able to detect Mycobacterium tuberculosis, and we later found that the patient was recently vaccinated with BCG. And there was a pleural fluid where many neutrophil, no organism seen, but by metagenomic NGS were able to identify strep pneumo or candida in this type of specimen. So then in the prospective study, as I said, which actually we recruited patients and subjects from all hospitals, tertiary care hospitals in Qatar. And then using the referral criteria, we received a specimen for over 5 years. And we tried to run, whenever possible, both Sanger as well as metagenomic NGS, but not necessarily all samples were sequenced with Sanger. There are a total of 187 patients with their age ranging from 0 to 90 years. But our patient population had a very small proportion of immunocompromised population, which was about 9%. And specimen tested by Sanger were about 180, and specimen tested by metagenomic NGS was 95. So in terms of diagnostic yield, using all this type of specimen, you can see CSF mostly from meningoencephalitis or encephalomyelitis, tissues mostly from endocarditis and lymphadenitis, blood and bone marrow from fever of unknown origin. We also did a bunch of brain abscess samples, a few synovial fluid from cases of osteomyelitis and septic arthritis, and pleural fluid, obviously, from empyema and pleural effusion. So the Sanger, about 180 samples, 51 were positive with a detection rate of about 28.3%, compared to metagenomic NGS done only on 95 samples, out of which 20 were positive, with a 21% positivity. Interestingly, the rate was much higher in the immunocompromised, although we can confidently tell because the sample number is very low for the immunocompromised. So this was the 2x2 table for Sanger versus metagenomic NGS. And so you can see that the sensitivity of metagenomic NGS was quite poor, like 70% compared to Sanger. And out of pathogens, that were detected only by NGS, obviously there are a few viruses which we do not expect to be detected by NGS and by Sanger. But the other 3 species of bacteria or fungi, there are 3 detected by metagenomic NGS, 3 are only detected by Sanger, and which could really be related to the low quantity of the target in the specimens. So now, what lessons we learned from these projects? First of all the challenges that we anticipated with shotgun metagenomic approach still remains. One of the most important thing is the presence of host DNA. If I give you the average data from our projects, as you can see, we had with the Illumina MySeq, we had per run about 18.5 million NGS reads per run. Of which only 0.19%, that means 99.8% is actually host DNA. So that is one of the biggest challenge in shotgun metagenomic approach. And we also attempted a lot of host depletion and enrichment approach, which is I'm not going to talk today, but I would say host depletion approaches that we have tried not universally works for all specimen type. So there are other challenges there. And also the cost is also high. However, the bioinformatics, which was a major challenge for us like about 12 years ago, there's a lot more new bioinformatic pipelines are now available. However, the test interpretation with metagenomic NGS is still challenging. And then you need actually some person with both clinical knowledge as well as technical knowledge, in particular experience with metagenomic NGS to actually appropriately call the results. If you go to the literature, you would probably see hundreds of successful case reports on the successful application of unbiased metagenomic NGS. But those papers doesn't reflect actually what is the actual diagnostic yield and cost of the test. So our study at least showed actually a poor diagnostic yield of the shotgun metagenomic approach, and our with a cost of about $2,000 per test. So that's why as Andrew also diverted his approach towards more targeted approach. We're also going towards more targeted metagenomic approach, such as again 16S 18S given us a great opportunity for bacterial and fungal detection. And also with targeted approach, we may be able to reduce the cost and simply improve the sensitivity and simplify the workflow of the laboratory. So for targeted approach, one is the amplicon sequencing, amplification-based sequencing, and also the bait capture method that Andrew has discussed as well. So we have proposed that we can probably how we can transition from shotgun to targeted approach using the 16S, 18S, or all kinds of universal target assays for bacterial and fungal detection. We could try to do a one-pot amplification and can reduce the sequencing cost is always cheaper when you can pool a bunch of samples and do barcoding. But in acute care setting for sterile site infections, it is most of the time it is a low volume test. Most of the time we would have to run one sample or two samples. So we have to take an account of that when we calculate the cost. So in that way, the cheaper flow cell or reusing nanopore flow cells is an option. Or Illumina also has a new version now, MiniSec, offering about $100 to $200 test. So based on this approach, we actually already have some proof of principle data generated at HRMP St. Joe's Research. We tested about 29 samples bronchoalveolar lavage from immunocompromised patients with lower respiratory tract infection. Of course, all the tests were negative in these samples, including viral PCR, bacteriology, mycology, mycoplasma, fungal PCR. So with 16S, 18S nanopore sequencing, we were able to detect 11 positives out of 29, which is nearly 40%. So that's a very high proportion of positivity. And the results, if you look at some of the organisms here, it makes sense. These organisms are not routinely detected by culture, like nocardia, metamycoplasma, trophirema, or a bunch of anaerobes. So these look like clinically relevant, given the limitations of culture and given the patient population. So all these samples were sequenced actually in one nanopore run, but in real time we may not have the opportunity to run all them together. So that's we have to consider. Other ongoing sequencing-based projects going on in our lab right now, we are still continuing validation of 16S ITS HSP65 for mycobacteria sequencing, because we want to do both Sanger as well as nanopore-based targeted metagenomics on the same set of samples, and eventually assess the cost, the resources, and the potential clinical benefit, and to decide about the right workflow for these approaches. Also, we do time to time Sanger sequencing for individual viruses, to look at, to find their serotypes. For example, an enterovirus surveillance project we did, showing a significant expansion of a particular strain of Coxsackie virus last winter. And of course, many of our projects right now are amplicon-based targeted. We are working on a nanopore-based CMV drug resistance assay, targeting 4 target genes and giving us resistance to several antivirals. And then we have one project going on. One of our postdocs had developed a really a nice sequencing-based serotyping assay, culture-free serotyping assay, which can actually resolve 60 serotypes and 22 pairs of serotypes without culture from crude specimens. And then we also worked on a little bit on the influenza A surveillance, which just to gain some experience on this. We have subtyped about 30 samples with about half H3N2 and H1N1. We did not fully give up shotgun sequencing because there are certain areas, certain objectives that you can fulfill by amplicon sequencing. So we tried a few bacterial whole genome sequencing. We sequenced 2 outbreak clusters. The MLST results we get from sequencing are consistent with PFGE, but the whole genome sequencing also gives us, allows us to analyze the plasmids and to go higher resolution genotyping by SNP genotyping. So in future, if it requires, we may be able to get ready for this kind of thing. And then for the viral metagenomics, especially, we started with a respiratory viral surveillance. We are facing a lot of challenges with host depletion, enrichment, and pool approach, but we are currently working on it. So majority of these are nanopore based. We would not say that all problems can be resolved by nanopore. We may still need to go back to Illumina for certain projects. And finally, in terms of antimicrobial resistance, Andrew already talked about the brilliant work they have been doing with the bait capture. And we hope to, and we plan to collaborate with him as well to do some hospital-based work using for emerging antimicrobial resistance, as well as planning for, probably a customized universal AMR screening assay for infection control. So in conclusion, DNA sequencing still holds a great promise to improve the diagnosis of infectious diseases, despite the many challenges we have identified. I think choosing the right patient group, right specimen, and right sequencing approach is critical to the successful application of DNA. And no wonder there's a lot more to do to standardize these methods and assess the diagnostic performance and clinical values of these tests. With that, I'd like to say that here, it's at HRLMP St. Joe's Research, we are actually want to work towards establishing a core clinical microbial genomics and metagenomic facility with all these different arms. And we would like invite anyone from this audience, to collaborate with us or to collaborate in funding applications. And I would like to thank you all, and I would like to thank a lot of people, just listed a few here who are directly involved with the work we have done so far. And thank you very much. We're happy to answer any question.
Thank you. Thank you both for those great talks. Super interesting. So we have time for a couple of quick questions. We have 2 from Mike Surratt. So the first question is for both of you. He says, thanks for these great presentations. There's a large amount of genome data in clinical and government labs that's not publicly available, which is an obstacle to academic research. Is there any discussion about making this data publicly available?
Rubayet, do you want to go first?
I think you'd be the right person.
Okay.
I am very new in Ontario.
Perfect. Yeah, so yeah, I'm pessimistic of the historical data ever coming out of these silos. I'm much more optimistic going forward. Countries like UK, Denmark, Netherlands are doing very well. This Canada traditionally has not, like the pandemic, Canada got pointed at for really slow at sharing coronavirus sequences online. And in the first year, McMaster was the leader, and we were a tiny sequencing shop compared to the others. So, and I'll Alberta Health Services were also very good. Yeah, and so what I've seen, I work with a lot of public health agencies in Canada. Federally, I see with all the open access and open data acts that have come across, all the federal initiatives I'm involved with, they're already dumping their data into GenBank. Like they've made that switch. That's part of their business plan, that as they sequence, it goes public. As I said, all those traditional historical ones, maybe not. Provincially, it's a dog's breakfast. Some provincial agencies are very good at it, and others are not doing it. A lot of it's just straight out funding, right? And they don't have enough bioinformaticians to do that. Ontario in particular is tricky because we're a federation of many health systems that may or may not be required to share their data with Public Health Ontario, depending on the pathogen and the question. And they're under-resourced, and unless it's a research aim, like if it ends up in a paper, then it's going to go public because that's how it works. But it's a real challenge. So I don't think it's hitting close to the local hospital that is doing sequencing. I think they were forced to do it through coronavirus, and it's worked very well, but it was a new experience for them that, oh, not only do we produce it, but we have to, it's a lot of effort to format and submit it to a database. But Public Health Ontario do well on some. They do go on coronavirus, but that's because they have agreement to share with the feds. Canada's federation, the laws of data sharing between province and federal are a real barrier. Our legislation's behind on this, but federally, I'm much more optimistic. Yeah.
I feel like the sequence technology has outpaced the storage and sharing aspects of this.
Yeah, you can point to, if it's outside of academia, you can point to any agency or hospital system, and they are under-resourced for the hardware, for sequencing, for the bioinformatics, but the people too is an issue.
Mm-hmm. Well, and I also take your point about, the physical moving of data from one place to another using infrastructure that's of various antiquities.
Yeah, absolutely. We do that every Wednesday with Hamilton Health Science, you bet.
Yeah. Okay, and last quick question for Rubayet. Why not sequence 16S amplicons by Illumina, which resolves mixed sequences and requires very shallow sequencing?
Yeah, that's actually a very good question. So I think with Illumina, I think we would probably have the biggest advantage is that the library preparation for 16 sequencing. So we can actually probably directly run the amplicons if we design our primers with the barcodes directly, right? So that's one. But Illumina, again, with nanopore, we can actually, which is really good in long read sequencing, and because we think about a lot of factors here, like we think about cost, like Illumina flow cells are not reusable. I still don't have experience with the mini-seq and with the minimum flow cells, how they perform. That's something we may want to try at some point, actually. So the cheaper one, but at least with MySeq, I know, with MySeq, our cost was, including library preparation, was nearly $2,000 per run. So with amplicon sequencing and no need for library, it would still be somewhere about $1,000, I think, or even more. So that is very expensive if you have to run a very smaller number of samples. So that's why, my plan was to using Flongol actually, which is very cheap, but Nanopore suggested that maybe the running and MinION flow cells, reusing them is better than the Flongol in terms of, because Flongol has very few pores, and probably if significant proportion doesn't work, then you would end up with a failure. So we haven't tried Flongol, but we have tried reusing Nanopore MinION flow cells, which works very fine. And Nanopore is also, so easy and handy to run, and it's very simple and very low capital cost requirement for nanopore. So yeah, but I think that question would still remain. If I would be interested on the mini-seq version of the sequencing, if we can try one day and compare with nanopore, that would be something interesting.
Yeah, thank you both. We're out of time, so even though I have a whole list of my own questions, we'll have to save that for offline. So thank you again for your talks. Really super interesting topic and very timely. And thank you everybody for staying with us through the hour, and we'll see you in November.